Good morning, Dogg from Air fans. Now, I have had some coffee, which is not something I usually do anymore. So this video is a lot of fun for me to research, and I've got to get it out of my brain before it explodes. All right, and so I am very pleased to put this video in my general category, not specifically in my COVID-19 series, which has been pretty popular. But it, of course, does relate to that, so you'll want to cross-reference them. At any rate, video is about the scientific problems with the field of virology. And there are quite a few that I don't mention, but this is enough, I think, for a layperson to hear and be like, that doesn't make sense. Virology is based on some faulty premises. So, first of all, a brief outline, there's 3 points. First of all, viruses have never been isolated. Second, viruses have never been demonstrated to be transmissible. And third, the study of viruses depends on immortalized cell lines in vitro. In other words, they grow viruses in cancer cells that have been found to survive outside of their hosts. So let's go to the first little thing. Never been isolated. Yeah, of course, everyone will tell you that they have. They do it all the time, and coronavirus has been isolated from patients, etc. But the question is, what does that really mean? Okay, and because viruses cannot be, do not exist by themselves, they cannot be isolated. So, like, you can go to the store and get a little jar of Fleischmann's yeast that contains those little pelletized, yeast spores, and you can use that to grow bread or to leaven bread, etc. You simply cannot do that with a virus. And a little bit into the history of that, okay? So the first virus to be quote unquote discovered / isolated, if we're being charitable, would be the tobacco mosaic virus, which was discovered, they say, by Martinus Beijerinck, who in 1898 independently verified the earlier results of Dmitry Ivanovsky. And what those fellows did, is they took some sap from a diseased tobacco plant and they filtered it through the smallest pore size filters they had available, resulting in some filtered sap, some fluid, which they could then inject into a healthy plant and get the same signs of tobacco mosaic virus. So in other words, they took out all of the particles they could find and they were left with a fluid. So the original definition of a virus, it's important to note, was a fluid. They didn't know whether there was something dissolved in it, or whether there were even finer particles that were the cause of the illness in question, which is the current prevailing theory. In other words, viruses are these particles that are very, very small. That is the current theory. Further, in 1935, a man named Wendell Meredith Stanley crystallized that sap and then found that the crystals, which were further reconstituted back into a liquid, could cause the same symptoms in another plant. So that is super duper interesting. But the question is, are those crystals, are those crystallized viruses? So what they're saying is that the crystals contain the viral RNA and the capsid protein, which is, viruses consist, they say, of the genetic material and then the wrapping around the genetic material. And then in the presence of the host, what they say is that this crystal, which has been dissolved, is able to reconstitute itself back into the viral capsules, okay? The viruses, the virions, the viral particles, that's what I'm looking for. So the question is, can they, okay? Do they do that in just water? Well, no, they only do this in a host, okay? So you only find these tobacco mosaic virus particles when you take this tobacco mosaic virus crystal and put it into the host of susceptible plants, okay? So that's a really big question. Are viruses alive? Are viruses alive? And of course, I mean, if you read about it anywhere, you'll see that they are on the edge of life. We don't know, okay? That's just what they say. And we don't know what life is. So I've talked about the metaphysics a little bit about that. But that's where viruses stand in our current scientific understanding, is on the edge of life, and we don't know what life is. So that is problematic, no matter where you fit on the materialist spectrum. And contrast that with the work of Antoine Béchamp and his microzymian theory, where he was able to measure the enzymatic activity of these particles that he called microzymes. And he was also able to measure their ability to reduce dissolved oxygen in water. So, therefore, proving that they were able to have some effect on their environment that would be persistent, and they would also be capable of reproducing, forming into bacteria, et cetera, et cetera. Anyway, moving on, real humdinger with tobacco mosaic virus is that it's never been shown what the means of transmission is for this virus, okay? So what they do in the studies is they take the sap and they inject it into another host, okay? So how does that relate to nature, okay? How would that thing happen in the real world, okay? And the citation for this is just going to be a gardening website I found, but it's got a ton of links to different university, ag extensions. So you can see what the current thinking in this is how it's evolved since 1898. Okay, this is just, these links are current. Go ahead and check them out for yourself. They found that it can be injected. They found that a person they think that a person, if they've been smoking cigarettes, they can transmit that mechanically by handling a plant, but it's unclear whether insects are capable of transmitting it, okay? After 120 years of research, they still don't know conclusively if an aphid can have the virus on its mouth parts and inject it into a host, which would, of course, be the analog if we're expecting a similar mode of transmission to the scientists in the lab injecting the purified serum into the host. Follow me? That's bizarre, but that's where we're at. The same is true with human studies into viruses, okay? So I'm going to include a link to a kind of roundup of all the ways they tried to test transmission of the Spanish flu back in 1918. And it's only a 3-page article, so it's easy to get through. But they did, all of the above. They injected stuff from patients into healthy people. They had the patients sit across from a healthy person and breathe into their mouth for 5 minutes, and it never worked. Basically nothing that they tried to do to get the Spanish flu into a healthy person ever worked. Okay, so that is very suspicious. And in fact, when they I think it was when they injected it, they actually found, they found that they had sore throats. They thought that they had the Spanish flu, but they ended up having a sore throat, which was caused by a bacterial infection, which they were able to isolate using their methods. Okay. So another thing that they have done. So first of all, this is called experimental transmission, or transmission studies is the field that you're type into your favorite search engine to find information on. And I would really like it if someone would push back at this and show me where I'm wrong here, because I haven't found really anything. I'm going to attach a link to a video where they're showing a mouse subject that they're using to test this transmission theory, and watch the video and see what they do. Briefly, they anesthetize the mouse with ketamine, they knock it out, and then they take a syringe full of snot, we would say, and fill the mouse's respiratory system with this fluid, okay? And they say in the video that if the mouse wakes up or if it coughs up some of that stuff, they have to use a different mouse, or they have to knock it out and keep getting more into it so that it sits in that mouse's lungs for a certain period of time. And my question is, how does that relate to like breathing stuff in, okay? How is breathing particles in similar to having your lungs flooded with fluid? And is having your lungs flooded with fluid by itself going to have a deleterious effect on your health? I think the answer is obviously yes. But so historically, there was this cool device they built at Fort Detrick, Maryland, that they used for studying aerosolized agents. It was called the 8-ball. You can look that up on your own. But this was an opportunity for Seventh-day Adventists during the, I guess, World War II and other periods of conscription to serve out their terms in the United States military as conscientious objectors. So they didn't want to fight on the battlefield, and instead they would take these guys and hook their faces up to this giant metal sphere, and they would pump various things that they wanted to study into the top of the sphere, and then there were fans and such, and they would suck it in and see what effect that would have. Okay, so that's all kind of like black project kind of stuff. So it is hard to figure out what the heck they learned from those studies. And I will grant you that maybe they found that these viruses, after satisfying all Koch's postulates, etc., were able to infect these poor Seventh-day Adventist volunteers. But please scream at me with that stuff if you can find any data there. I'd love to hear it. All right, so how do they study viruses? This is where it gets really wacky. So first of all, I've got to go back and explain something called Koch's postulates, which is a set of requirements for demonstrating scientifically that a pathogen is the cause of a certain set of symptoms in a host. So the Koch's postulates were written down by Mr. Robert Koch in 1884, and I'll go through them briefly. The first is that the microorganism, or in this case, substance, is found in abundance in diseased organisms, but not in healthy organisms. So right away, this disqualifies COVID-19 as a viral illness because the viruses are found in many healthy patients. In other words, we have the asymptomatic transmission. Okay, we have people who test positive via the PCR test but never show symptoms. And this demonstrates that at least in those patients, the presence of the virus or whatever they're finding cannot be really the presence of the DNA or the RNA cannot be considered diagnostic for the coronavirus, the COVID-19 illness. That is true, though, for the tobacco mosaic virus, where these crystals, and that's why I use the word substance, these crystals were found in the diseased plants. They were not found in the healthy plants, okay? Number two, the microorganism is isolated and grown in a pure culture. Now, this is just impossible for viruses, so that's out of the question here. Number three, the cultured microorganism causes disease in a healthy organism. So this is where we get into problems with the whole of germ theory. How do we prove that this organism causes the symptoms that we are seeing? And finally, the microorganism is re-isolated and identified with the original. It's shown to be the same as the original organism. So what they're saying is that the Koch's postulates are inapplicable or need to be modified when applied to to virology. And to that, I'd say, maybe, but how do you demonstrate that? I'm going to include a link to a fun little study that I mentioned in an earlier video that is talking about that, with specifically addressing how Koch's postulates must be amended to viral culture. And I might add here, the observed properties of a virus depend entirely on the host in which they are cultured. So, how do they culture viruses? Because remember, viruses, what they're saying is they don't propagate outside of their host. And this is why we're saying they're on the edge of life. Okay, they can supposedly move around, etc., but they can't reproduce, and that's one of the conditions for being alive. So for vaccine production, they culture them in animal cells, or, well, historically they have anyway, and that means that they will take some monkey kidneys or some chicken eggs or whatever they find is effective, and they will blend them up and put the viral or whatever stuff into that, and then they'll cook it for a while, incubate it for a while. And not only that, but they have to go through a very specific set of what they call passaging, where they will go through 16 times through monkey kidneys, 4 times through fetal bovine serum. Well, that would not contain cells anyway. A couple times through a chicken egg. I mean, I have newt. It's very specific to each virus or each disease that they're trying to culture. Very, very specific, which is why this is so difficult, and it's all happening like in these ivory towers with massive sources of funding. But on the other hand, for growing viruses for study, they use what are called immortalized cell lines. Okay, so first of all, normal cells in our bodies will stop dividing after a certain period of time, and that is why we die, which is bad. But there's a set of trade-offs there that I'm not going to get into. Immortalized cell lines, they found, for some reason, do not divide, or do not stop dividing, excuse me. So in other words, obviously what immortalized cell lines are cancer cells that they have found growing in various hosts, and they have taken them out of the host, and they have found that they actually grow in a test tube. Okay, so they are extremely resilient little buggers that they have found, because most of the time too, you can't just take a cancer cell and put it in a petri dish, with growth medium, and expect it to proliferate. Most of the time, they're just going to die. So these ones are, a select few cell lines that they've managed to get from the wild, and they've basically domesticated them. Okay, so in other words, immortal cell lines are domesticated cancer, and that is what they use to study viruses. So how many of those are there? There are 124,601 immortal cell lines from a website called Cellosaurus, which looks pretty cool. I'll include a link below. But one of the really interesting things about these different immortal cell lines is that over time, when they got into DNA testing, they found that where they thought they had a bunch more immortal cell lines, many of these immortal cell lines that they thought they had actually been infected by others. So there's certain immortal cell lines that are just incredibly strong, basically able to live in a test tube or a petri dish, and they actually will somehow get into these other ones that they thought they have, and grow there, outcompete them. Also, fun fact, they have injected some of these immortal cell lines into immunocompromised prison inmates and found that, yes, they are capable of growing in other people. So, in other words, they're going and they're finding, cancer cells from a certain organ. It might be like, in some cases, it's going to be uterine cancer. In the case of Henrietta Lacks, different kinds of cancers. In the case of COVID-19, they are using the lines of Vero CCL81 and Vero E6. These are the immortal cell lines, the cancer cells that the CDC uses to grow coronavirus. And they were isolated in 1962 by a Japanese scientist. And what they are is they are epithelial cells from an African green monkey kidney. They're kidney cancer cells is what they're using to grow coronavirus. So what are the problems with this? Okay, first of all, it's cancer. They are growing viruses in cancer, which has an abnormal number of chromosomes, it has abnormal mitochondrial activity, on and on and on. And what can we learn about a normal body by studying how these things proliferate in a cancer cell? Further, there is no immune system in vitro. In vitro means in glass. It refers to a test tube or a petri dish. Okay, the cells are on their own. It is just a pure culture of cancer cells, with no blood, no white blood cells. So the way that our bodies fight off invaders is not there. Okay, so therefore, what are we even learning? Okay. Yeah, the cells aren't, there's no aerosols, okay? The cells aren't breathing in, there's no lungs. The viruses aren't being injected, there's no blood. There's no temperature fluctuation like occurs in a normal body. There's no nutrient variation, there's no hormonal variation. All of the different factors, which admittedly are very hard to study, and that's why, the science tends toward these, reductive approaches, aren't there. So again, what can we learn? Okay, viral culture in immortalized cell lines is not sterile. That means that the cells themselves, because you can't kill them, you can't sterilize the cells, that would kill them and that would be ridiculous because you need them to grow the viruses. The cells themselves may be bringing with them, viruses, such as was found to be the case in SV40, which is linked to the Cutter incident and the mid-century polio epidemic. And finally, the growth medium that they are using is not inert. So they are not just growing viruses in a petri dish with cancer cells and nutrients to grow the cancer cells. They are also including antibiotics and antifungal medicines in that medium. And those are bioactive compounds, which means that they have an effect on those cancer cells, which is studied and known. And they have to, that's a confounding factor in saying, like, are these particles possibly being produced as a result of an interaction with the antibiotics, or are they truly being produced as a result of this thing that has, is being put into the test tube with the viruses and the growth medium, which again is not isolated in the first place, okay? When they grow viruses, they aren't starting with an isolated, viruses only that's going into this mix, okay? Just how it works. So, to wrap up again, they've never been isolated. Viruses have never been demonstrated to infect healthy organisms. And finally, virology is dependent on the effects observed in cancer cells in a totally unnatural environment under very specific conditions. And the question has to be asked, what is the application of this field of study to healthy or even diseased organisms? So that's it for me. I'm going to close this one out. Please disagree with me. Please give me some pushback. Please give me some data. And please share this video. Comment down below, like, and subscribe to my channel. Thanks so much for listening. This has been Evan Benton of Dogg from Air. Thank you, and have a Merry Christmas.